Please go ahead and we look forward to your talk.
Thank you, and thank you very much for having me, Adil, and for organizing the seminar series. What I'd like to talk to you about today is self-assembly. And, what is self-assembly, right? So the idea is that you take a bunch of objects which have certain shapes, and very, very, very clever people have thought about this before, and actually evolution has thought about it before. And in many cases, inside the cell or in artificial settings, what happens is that those objects fit very nicely together and come together in order to make a structure which is functional or interesting in some way. Now, the topic that I'd like to talk to you about is something that is not so smart. That is, you don't consider objects that fit nicely together, but you ask something different. You ask, what happens if I have objects that are mismatched in some way? What if I have puzzle pieces that don't quite fit together? And what kind of shapes can they form? And what we claim is that in some cases, they actually form objects that are quite predictable, despite the unpredictable nature of the misfits between the different puzzle pieces. And those structures might end up being slim aggregates, fibers, or planes in 3 dimensions. And the basic idea for this actually comes from a biomedical setting, and specifically the case of many diseases in which you have proteins that are normally soluble inside the cell, meaning that evolution has not optimized them to fit together nicely, but due to some accidents, they start aggregating. And those accidents can be a misfolding, mutation, for instance. And when they do start aggregating, the surprising thing to the physicist is that they don't form a crystal, they don't form an amorphous 3-dimensional clump, but very often they form fibers. And that is valid not in 1 or 2 cases, but across many different types of proteins involving many different types of molecular interactions between them. So the idea for us is not to go and question the detailed molecular studies that people have done about this fiber or that fiber and the detailed molecular understanding that they've gained on those systems, but rather to take a step back and ask whether there might be a generic physical effect that is responsible for the formation of these fibers, and that is valid across a wide range of molecular details. And now, in order to explain what that principle might be, I actually don't know any better solution than to wave my hands around. So I'll ask you to bear with me a second as I turn off the shared video and enlarge my picture on the screen if you want to see what I'm doing with my hands. But the basic idea is that if you have irregular objects, they might have valleys and ridges, and they might have some places that are more sticky and some places that are less sticky. And so if you take several copies of those objects, they happily diffuse in solution, and when they find each other, they will try and go in the most advantageous configuration, relative configuration that they have, but that forms the first dimer. Now the question is, what happens when you come in with a third particle, which has this complicated shape that does not specifically fit together with the other 2? Well, essentially, there's at least 2 things that it can do. And the first one is to come in and try to take advantage of that best binding site that was inside my hand here and deform the existing aggregate to participate in this best binding. That's the first extreme of the spectrum. And now the other extreme is essentially a situation where the aggregates are not, the proteins are not too deformable. And so the new particle comes in and sticks at a different location, which is perhaps the second best location in terms of binding. But in any case, that second binding event is going to be less energetically favorable than the first. And as you add more particles, essentially the situation is going to become more and more constrained at every step along the way. More and more directions are going to be forbidden. There's going to be more and more steric hindrance between the different objects. And so at some point, you could imagine that the energy bonus that you get for aggregating becomes less and less. And maybe at some point, it's so bad that it's not worth spending the chemical potential of the solution in order to bring in a new particle. Now, that argument essentially tells you that maybe you might get a mechanism for limiting the size of your aggregates, but it doesn't specifically tell you that you should be making fibers. And so I'm going to throw in one more assumption, which is or speculation, which is that essentially fibers are actually not such bad objects in this competition between adhesion and frustration, because they're essentially large objects. So there's a lot of contacts within them, which is good for adhesion. But at the same time, their diameter is limited, meaning that every particle within them is close to the boundary of the aggregate. And the boundary of the aggregate is where you relax your frustration. It's where the particles have more freedom to choose their orientation or maybe deform in some direction. And so for that reason, maybe fibers could be a good compromise between those two basic energies that I'm going to come back to over and over again during the talk. And what I mentioned before is that essentially there's two things that the particles can do as they come together. And I'd like to highlight them in two separate models during the two first sections of my talk. The deformation on the one hand, and then those anisotropic interaction, this idea that there's some binding directions that are better than others. Then, for those of you who are a little bit introduced to those topics, I'd like to talk about amyloids, which is a very widespread model for how fibers might form. And I'd like to, discuss how what I'm proposing is different and maybe a useful complement to the notion of amyloid formation. And then the last section will be about something more speculative and tentative, which has to do with trying to see indications of this fiber formation in some actual protein aggregation data. So let me start with the deformation model at first. And essentially our notion of a particle in this case is extremely simplistic. We think of particles that are polygons in 2 dimensions. And there's 2 types of polygons. There's the good polygons and there's the bad polygons. The good polygons are the ones that fit nicely together and you can tile the wall of your bathroom with them. And indeed, if you put them together, you can essentially cover the whole plane without incurring any frustration. But that's actually an extremely specific case, and generically polygons do not behave that way. If you take, for instance, a hexagon that has irregular sides, when you try to fit it together with its friends and you demand perfect contact between 2 particles, what you're going to have to do is to deform those particles a little bit. You see that this one is expanded horizontally, this one is squished a little bit. The whole thing is starting to develop a little bit of a bend. So you have to torture your objects a little bit in order to make them fit together. And those are the ones that we are going to be concerned with, the ones that require a deformation if you want to pack them tightly. So there's 2 extreme limits of the behavior of those systems that are very easy to understand. And essentially it's governed by this comparison of the 2 energy scales that I mentioned before, adhesion versus frustration. And essentially what I've indicated at the bottom of the screen is the adhesion energy, the surface tension of the aggregate in units of the frustration energy. If the surface tension is very large, essentially you're always going to be able to torture your objects to a finite amount that will make them fit into a crystal. And so you're going to form a bulk. Now, if you have a very low surface tension, that means that the adhesion is a second thought, and your most important constraint is that you want to maintain the ground state shape of the particles at all costs. And so one thing that you can do in this case is, for instance, put your particles together into that tree, which has an enormous surface area. So in a way, if the surface tension was large, this would be extremely costly. But if the surface tension is very low, then the aggregate is happy because each particle has its ground state shape. And the speculation here, which I mentioned before, is that there is an intermediate regime of intermediate surface tensions where fibers might generically form. And the reason for this would be that fibers might be a good trade-off between stickiness and frustration. So let me get a little bit more specific about the model that we're actually using. The energy of the aggregate is the sum of the energies of its constitutive particles. And the energy of a particle is the sum of 2 terms. Again, they correspond to those two energies that I've mentioned several times before. And in order to understand the deformation energy, you have to think of each of those hexagonal particles as a rubber balloon. So why is a rubber balloon rigid? A rubber balloon is rigid because it's essentially a membrane that wants to shrink down to a point, but it can't because I blew gas into it. And so the gas tends to expand, and that tenses the surface of the rubber balloon. So in this case, the role of the gas is played by this term here, which essentially gives you a lower energy if you have a larger area. And the role of the membrane is played by a sum of harmonic springs with 0 rest length, which correspond to each of those sides. If I want to make a regular polygon, then all those Ks would be the same. And in that irregular polygon, I've essentially taken a different spring constant on the odd and even numbered sides. So that's for the elasticity of an individual particle. Now the stickiness is again very simple. I just count the number of sides that are actually exposed to the outside of the aggregate, and I multiply this number by a certain parameter sigma, and sigma is the surface tension that I mentioned before. Now, once you've decided what the topology of your aggregate is, so the adhesion, the notion of adhesion in this model is again very simple. It's an all or nothing notion. So two sides are either completely joined together. That means that their two vertices have exactly the same positions, or they're completely disjointed, right? There's no notion of a range of interactions, a zero range, all or nothing interaction. And so that's a notion of topology that you have to introduce, right? Connected or not connected. And once you've set the topology, of course, you have a freedom to set the position of all your vertices in the elastic network, if you will. And what we do implicitly in all the following is that we use the computer in order to minimize the elastic energy at fixed topology with respect to all those positions, which is the same thing that as saying that every aggregate that I'm going to show actually has force balance respected throughout. All right, so what's the result of this model, right? And here I'm going to show you an example where I start from a single pentagon. And what I'm going to do is I'm going to take another pentagon and I'm going to try to stick it to the first. And there are several ways to stick it. I can stick it to the first face, second, third, fourth. Of course, they're all symmetric at this stage, but it's going to change later. Or I can even, try to stick not two faces together, but 2 plus 2, right? 4 faces. So what the computer does is that it computes the energy associated with each of those solutions, and it chooses the most favorable. So it's a greedy procedure. At every particle addition, it chooses the best solution for the next step. And here's what it looks like. So at the beginning, it doesn't look like much, right? You don't know where this thing is going. But at some point, for some reason, it catches some kind of an attractor, and it starts going on and reproducing this periodic pattern that grows forever. And the mathematical definition of forever is long enough for my audience to get bored, so that clearly completes the proof. So now why does it happen? Well, the real answer is I don't really know, but I have a little bit of a speculation that I can share with you. And the speculation consists in considering a small aggregate that would have a little bit of an anisotropy. And the question is, am I going to amplify this anisotropy, or am I going to make it less and go towards a sphere? And there's two basic options that you can do as you add a new particle to this aggregate. You can add it to the side, or you can add it to the tip. And each of those has its advantages. This one, for instance, allows you to tuck the new particle in existing particles, and so it has a relatively low surface cost. The problem is the shape of this particle is not quite matched with the shape of its neighbors, so it's going to deform all those neighbors, and because those neighbors are connected to further neighbors, the elastic deformation is going to propagate, and you're going to affect a relatively large region for a relatively large elastic cost. And here, of course, you have exactly the reverse benefits and costs. So if you start with an aggregate that's actually initially pretty small and pretty thin, it would make sense that initially this option would be relatively cheap because since your aggregate is so small, this elastic deformation doesn't propagate very far. It's essentially screened by the free boundary of the aggregate. And so you could add particles to broaden it for a while until, of course, your aggregate is not thin anymore. And when it's not thin anymore, this option is relatively more costly, and you will have more of a tendency to go here. And then as you add more and more particles to the tip, you're essentially ending up with your fiber. And essentially the mechanism that I've proposed here doesn't really depend on the shape of the particles. It doesn't necessarily have to be a hexagon. And indeed, when you try different types of particles, pentagons, irregular hexagons with different ratios of the size of the sides, or octagons, in each case that we've tried, essentially you get an intermediate tension regime where you get those fibers. So it's a cute phenomenology, right? But is it just the model is weird in all kinds of different ways. So is it just a peculiarity of the specific implementation? And in order to understand this, we try to have a model that we can understand analytically a little bit better. And the idea here is to do a different model that essentially has hexagons. And again, they're elastic particles. And what we say is that those hexagons have blue and red vertices. And the rule is the blue always goes with the blue and the red always goes with the red. Now, inside each of those particles, we essentially say that there's a bunch of harmonic springs. There's blue springs connecting the blue vertices, red springs, and also black springs connecting the two different colors. So when you put all those particles together according to the rules that I've mentioned before, essentially all the red triangle form a red triangular lattice. And there's also a blue triangular lattice. And one of them is larger than the other because the initial triangles had different sizes. So if you go to a continuum limit, it's as if you had two elastic sheets, one large and one small, and they're coupled together in such a way that you want to match the edges, but you also want to match the middle together and the 3/4, etc. Right? So the basic idea is if you have a very, very large sheet, essentially you're going to have to match their sizes. And so you're going to pay the elastic deformation of the blue and the red. But if you go towards the edge of the sheet, the red is going to be able to move with respect to the blue, and you're going to get more of the deformation of the black springs. And essentially, there's going to be a penetration depth of this deformation going from the blue and red shifted with a deformation of black to the bulk situation. And that characteristic length is going to control the behavior of your aggregate. And indeed, you can cast this in more mathematical elastic terms. And what you find is that depending on the parameters of your problem, namely the surface tension sigma that we have encountered before, as well as the Poisson ratio of the individual particles that we've introduced, you might push your system towards different types of aggregates, starting from something that's tree-like, as we have discussed before, all the way to a bulk, and going through a possible succession of just a fiber or a disk and then a fiber. And essentially, if you do Monte Carlo simulations of the model that I've presented before, so you don't just take Ansätze and minimize the elastic energy analytically, which was what the phase diagram was. What you find is that indeed, if you start from that low surface tension in the disk region, you start with small objects and get larger and larger disks. And here, for instance, you start from something that's clumpy, and then you get fibers that arguably are broader and broader. And yes, this one here is more disc-like, and then you go towards something that looks more like fibers. So it looks relatively reasonable, and we're starting to get a handle on how the system works. All right, so now let me move on to a different model. And the reason to do this is essentially to ask, does the other notion of frustration that I've mentioned before also bring about fibers? And in this case, I'm going to define a model that's completely different, and it's a model that lives on a lattice. And what I want to emphasize in the model is this idea that particles like to bind in some ways in some directions and in other ways in other directions. So I can say that my lattice is a triangular lattice, and each of my particles is a Voronoi cell of that lattice, which is a pretentious way of saying that those are hexagons, right? Hexagons have 6 vertices, and each of them is colored blue or yellow. And the physical rule is that blue likes blue and yellow likes yellow. So let me take that particle and put it on the lattice. Okay, that's fine. Now, if I want to bring in a second particle there in order to match the blue and yellow vertices, what I'm going to have to do is to rotate it a little bit. And so I'm going to get that situation. I'm going to have 2 sites that are color matched. And so I'm going to say that my energy is essentially -2 for 2 satisfied sites. But in doing this, what happened is that I created this yellow pocket of 3 consecutive yellows. And of course, there's no 3 consecutive yellows on the new particle that I'm taking from the reservoir to put it there. So I've essentially limited the growth in some direction. And at the end of the day, that's where the frustration comes from in this system. That's the reason why I'm going to limit the growth of my aggregates in some directions and not others. And nevertheless, what the computer can still do with some metropolis rule is to take that particle, rotate it, and put it here, or even start a new aggregate that's disjointed from the first one. But unlike the previous algorithm, which was a little bit weird because it was irreversible, in this system, I essentially allow the system to take back a move, again, with rates that respects detail balance. So if I wait long enough, my system should go to equilibrium. Okay, so now I've lied to you a little bit because I've told you that I'm looking at a 2-dimensional lattice. That's not the case. I'm actually looking at a 3-dimensional lattice. It's an FCC lattice, and the Voronoi cell is not a hexagon now, it's a rhombic dodecahedron. And the rhombic dodecahedron has 14 vertices. Each of them I can color in one of 2 colors, blue or yellow. So if I'm extremely naive, I'm going to say, oh, how many possible decorations do I have for my particles? I have 2 to the 14. But of course, that's way overcounting because some of those coloring are just rotated images of one another or mirror images. So I can just clean out all those redundancies. And at the end of the day, I end up with 288 distinct particles that I'm going to call molecules in the following. And so rather than ask which one of those particles is more representative of the physics of a protein, what I'm just going to do is that I'm going to take the computer and simulate all of them, see what comes up. And the simulation is essentially the following. I'm taking the particles and I'm putting them in the box at a high temperature, and then I'm slowly lowering down the temperature, simulated annealing, and the result that I should get should be some approximation of the ground state. So what I'd like to have in usual talks is to have people guess which one of the 4 particles that I presented here is going to give a 0-dimensional cluster, or a 1-dimensional fiber, a 2-dimensional sheet, and a 3-dimensional crystal. There's one of each in this video, but exactly, you can't do that because Adil said no talking. So I'm just going to play the video for you. And so that's what it ends up being, right? So what's the lesson that I should draw from this? The lesson that I should draw, and that you draw when I ask you, is that it's very difficult to guess. And we've tried, not just by looking at them, but having, ways to quantify the frustration associated with each type of particles. And the relationship is just not obvious. And it's something that we're looking into and trying to understand with more sophisticated means. But anyway, the phenomenology that you get is indeed quite diverse in those systems. And there's a significant fraction of the total molecules that I've presented before that will actually give you fibers or sheets. So the question is now, are those the actual ground state of the system? And even though they pop up in those simulated annealing simulations, what you can do is that you can actually enumerate all the different periodic structures that are accessible to each of your 288 molecules. And we do we enumerate those structures up to pretty big unit cells of the periodic structure. And we essentially look at 4,000 different types of structures for each of the particles that I've defined. And I think it's essentially an exhaustive search up to lattice size of 40 or 50, something like this. And what we find is that we can compare the energies of all those different candidate structures, and the fibers are never the ground state. However, what you can also do is to look at the bulk energy of each of those structures that I've enumerated and talked about, and you can look at the surface tension that's associated to it. And if you have a bulk energy and a surface tension, you can estimate the nucleation barrier for each of those structures. And out of this, you can reconstruct a kinetic scenario that essentially tells you, what if I start with a solution? What's the first structure that I'm going to nucleate? And then what's the second? What's the third? And what's the associated timescale? And you're essentially looking at the scenario right here. The 0 to 100% is all the 288 molecules that I've mentioned before. And essentially on very short timescales, if you run a nucleation experiment at constant temperature, which is not exactly the simulation that I've shown you before, is that you should first get low dimensional aggregates, 0-dimensional, 1-dimensional, but then over time, they will disappear and you're going to have sheets and bulks instead. But the devil is in the details, and essentially in the details of the horizontal axis, because this is a log scale of the nucleation energy. So it's a log of the time. So that scale goes pretty fast. Actually, the age of the universe for any reasonable assumption is somewhere here. So, okay, the 1D fibers are not the ground state structure, but a pretty good metastable states. And maybe that's a possible explanation for why they form. Now, there's other explanations that people have come up with in the past for why fibers form. And the most classical one is actually amyloids. And the idea of amyloids is that it's a universal mechanism in the sense that it applies to all types of proteins. And what you can do with proteins is unfold a little bit of their sequence, and put the protein backbone in contact with the protein backbone of another copy of the protein. And essentially what you form when you do this is that you form beta sheets that stack on top of each other because of mainly hydrogen interactions. And you can make a very, very large stack of this. And so provided you're willing to denature your protein a little bit, you can make fibers that look like this. So, do I need to even think about frustration and irregular objects? Isn't this the explanation for all things fiber in the biological, or pathological fibers, at least in the biological realm? Well, no, not necessarily. I mean, those things exist, that's for sure, but they don't account for everything. First of all, there are some cases of protein stacking into fibers that just don't involve any unfolding. Unfolding requires a fairly large energy, and you're not always doing this. That's the case of this one that's involved in sickle cell anemia. This one is involved in ALS. Another indication is that you can actually, if you have an amyloid, you can actually infer from the interaction between the different protein backbones what kind of mechanical characteristics you should get as a function of the width of your protein fiber. And indeed, all the compounds that are in the blue band here have a bending rigidity that is consistent with what you would expect for amyloids. But there's a lot of them, I mean, some of them that are not pathological, so they're selected by evolution, but also some that appear by accident, such as the ones that I was interested in, that are not consistent with this idea. So that's an indication that there are other interactions that matter besides the stacking of the beta sheets. Now, even within amyloids, people are increasingly discovering that things are not necessarily as simple as they assumed. And it turns out that just saying I'm putting one thing on top of another is not the final answer. And there's actually many different ways in which those things can come together. There's significant polymorphism, even in the more classical amyloids. And that argues a little bit in the direction of having this idea that you have, structures that don't fit together and, can arrange like this or like that. And indeed, having multiple ground states is a hallmark of frustrated systems. And then there's other proteins that are, other protein fibers that actually display the hallmarks of amyloid aggregation, such as the, this 4.7 angstrom ring in some conditions, but then at other temperatures, and actually for this yeast prion, this one is the more physiological temperature, they just aggregate in a completely different manner. So again, this is a complicated question, and there's not a single protein fiber structure, even for a given protein. And maybe the one that I like the best is that, people report the structures of the things that they can resolve, right? And all those amyloids look so nice and simple, and they are simple. And that's the reason why people were able to figure out exactly what their structure was. But if you talk to people in the field, what they actually tell you is that beyond those very simple examples, there's a wealth of other fiber forming aggregates that are much less well understood in structural terms. So, armed with this argument that maybe there are reasons to aggregate into fibers beyond forming amyloids, what we'd like to do is to go to actual proteins and try to figure out if indeed you have fibers forming in random aggregating conditions. And actually the best thing to do in this case would be to go and take 10,000 different proteins and hire 10,000 different postdocs, and each postdoc gets a protein assigned, and they're going to try for 2 years different types of aggregation condition, a little bit more depletant, a little bit more salt, and so on and so forth. And at the end of the day, they take many pictures, and we have a big group meeting, and we do statistics. How many fibers did you guys see over the past 2 years? But of course, that's maybe not the most practical plan. But there's a lucky break, however, because there's a whole community that is doing those exact experiments, although for different reasons. And this community is protein crystallographers. What they essentially want to do is to produce protein crystals in order to resolve their structure. And to do this, they take their favorite protein and they put it in those crystallography plates in many different type of physical chemical conditions. Again, soap, salt, depletant, and many other things. And they try to push them together, but not too hard, because if you aggregate them very violently, you're going to make something very amorphous and not nice and crystalline. So they try to put themselves in the same realm that we're interested in. And they're successful, rarely. Essentially, the success rate, if you have a plate like this, it's 1% of the conditions that you tried are going to be a crystal, or maybe even less. But the cool thing is that what they do in modern times is that they typically try all those different conditions inside those plates, and they take those plates and they put the whole plate under the X-ray beam in the synchrotron. And so the automated procedure essentially goes through all the wells and decides, oh, this one has Bragg peaks, this one doesn't have Bragg peaks, and the ones that don't, they throw in the garbage. But those are exactly the ones that we're interested in, right? The ones that didn't form crystals. And the hope is that the small amount of information that they got from scattering off of those things that don't produce Bragg peaks might be sufficient in order to find out what kind of an aggregate you were dealing with. Now, if this was, nice and clean sacks, of course, there's standard procedures in order to infer this, but this one is uncomfortable in terms of the place, the scattering angles that you're looking at, and so on and so forth. So it's a little bit of a challenge to try and extract the right information from them. And so what we did with our collaborators is to run, just as a proof of principle, was to run proteins that we knew formed fibers. In this case, actin and alpha-synuclein. It's not that they have anything to do with the mechanism that I'm, discussing today, but they're just well-controlled examples of fibers or no fiber, depending on what conditions you put them in. And what you find is that in this standard crystallography setup, you do get a little bit of a difference of scattering intensity, depending on whether you take a sample that has fibers, that has just monomers, or that doesn't have any protein as a control. So what you can do across, collecting a few hundreds of data points is that you can just ask the computer, can you, distinguish between those? And essentially the simplest thing that we've started to do is principal component analysis. And the interesting thing is that the procedure didn't know what was a fiber and what wasn't. But in this principal component plane, what we find is that the fibers go in some region and the monomers in some other region, and the controls in yet another region. And so even though this is not extremely intelligible at this stage, it appears that we have a procedure that would in principle allow you to distinguish between samples that are fibers or not. And what we do next is we try to understand a little bit, which features of the scattering signal actually encode this dimension of the aggregate. And so what we do is that we produce a bunch of fictitious aggregates inside the computer and simulate scattering off of them. And the question is, if you train a machine learning algorithm to recognize a fiber versus a crystal, will it be able to recognize them in a general situation that you haven't trained them for? And indeed, that appears to be the case. So when you look at the training set, for instance, you feed the true dimension to your neural network. And what you find is that in most of the cases, you're able to recover the dimensionality, the true dimensionality of the aggregate. So on the training set, it's of course very easy, but the test set is actually also comparably successful. So what we're hoping is to, take this little toy out of the computer and, recognizing simulated scattering and actually apply it to real data. And at the end of the road, of course, to apply it to the enormous amounts of data that the users of synchrotrons have produced over the years and that are in many cases publicly accessible. All right, so the bottom line today is essentially that if you make particles aggregate together that have strange shapes, they're going to incur frustration, and that might make the aggregate slim. And I think this has implications for, several questions. One of them is protein aggregation. Why do you form those fibers? And actually what I want to do is to reverse the question, because what I'm suggesting is that forming fibers is really the default option. So instead of having to explain why you form fibers, the thing that needs explaining is any case that you might discover that doesn't form fibers. Now, the reason why the collaborators, Monica Spano and William Shepard, whom I mentioned before, were interested in this project was not only to try and understand why those fibers form, but also because what we're hoping is that this might help guide users of the synchrotron towards proper protein crystallization conditions, which is the game that they're very interested in. And, if I had to daydream a little bit, I might say that the way in which an aggregating protein selects this or that crystal polymorph, or maybe no crystal at all, is really linked to the initial aggregate that it makes when it starts aggregating. And that initial aggregate could be a fiber or something else. And if we manage to understand what the initial shape is, that might help us understand the crystal that it then subsequently templates. And of course, you don't necessarily have to apply this to protein. In general, this should apply more broadly. And this could be a design principle. In other words, I told you in the beginning, right, that we're doing something that's not very smart. That is, we were taking puzzle pieces that are not adjusted together and putting them together. Essentially, what we're doing, in a way, is that we're not telling the system you should have this shape and you should make this structure. What we're doing is that we're taking self-assembling particles and we're asking them, what do you want to do? And then at the end of the road, it might be the case that they do something predictable and that we can then use this as a robust design principle. Thank you.
Just unmuting myself there, Martin. Just let me stop the recording.